Science in Pajamas, round 2. Alright, so today we're gonna, same PJs, it's the same day, it's all good. Now, today we're gonna talk a little bit about the history of DNA and how we came to discover it and its function. We're gonna keep it really brief, not gonna get into too much detail. Now, I know a lot of these names on the board, this is just round 1. My bio kids, you may not know those. That's fine. We didn't go into this much detail. But my genetics kids, I know that we talked about these because they're in your notes. So, ha! But again, just a really brief review on each one of them. So, start off 1869, and we have Miescher. And what Miescher figured out, hold on a minute. So, remember at this point, we didn't really know what DNA was. We didn't know its function. But this was the year, 1869, that DNA was first isolated. So they managed to actually pull it out of a cell, just like we did in our strawberry lab, if you remember, the DNA isolation lab. So it was much the same thing, that he was able to isolate and find this particular molecule. Now, again, didn't really know what it was, so he called it nuclein, Nuclein. All right. And then we skip ahead to 1902, where we have Garrod. Now, Garrod was a man who came through his studies and his research, found that a lot of disorders and diseases kind of involved defective proteins. Either the protein wasn't doing its job correctly or it wasn't doing its job at all. So according to him, he believed that it was a defective enzyme that was, since that's what caused the disorders, that it was our link to heredity. That the heredity molecule had to be linked to the proteins. He even published a whole book about it called Inborn Errors of Metabolism. Let's see. Then we move on to, I can't remember if I said it, so just in case, 1902. Moving on to 1928, and this is where we have Griffith. He started looking at DNA in terms of it being a transformative factor, how when you mix it with things, you can kind of see how different viruses or bacteria can change their shapes. We're not going to get into a whole lot of him, but we are going to talk a little bit more about 1944 when we have 3 gentlemen, Avery, McLeod, and McCartney, and they continued on with Griffith's work. So a lot of times when we talk about DNA as a transformative factor, we talk about them and their work because it builds off of Griffith and confirmed that it was DNA that was having this transformative factor. Well, they're the ones that showed that it was the DNA that was a transformative factor. Now, how they did this, they used a strain of pneumonia. And there are 2 variations of it, the rough strain and the smooth strain. Now, with the rough strain, when they injected it into a mouse, the mouse lived. It survived. But with the smooth strain, when they injected that into a mouse, the mouse got really sick and died. So then they thought, all right, let's kill the S strain, the smooth strain. So they killed the S strain, and then they put the dead bacteria into the mouse as an injection. And what do you know? The mouse lived. Then they thought, all right, R strain lived. He killed S strain, lived. What happens when we mix those together and put them in the mouse? Well, the interesting thing was, at this point, the mouse died. And what was discovered is that the living rough strain had absorbed something, had taken something from the dead S strain. And what that did was it actually transformed it from the rough strain into the smooth strain. So it went from non-fatal to a lethal strain. And that's when we figured out there was some kind of transformative factor, and it was DNA. Now, give me a moment while I erase this, and then we will talk about, all the other good stuff. Now, the next few names my bio kids will definitely have heard of. Maybe not the first one, but definitely the ones after that. So, give me one moment. See, look, a little bit of water, a little hand towel. Comes off a lot better, doesn't it? Alright, so clean. Goodbye, little mousey. Except for the blue. Maybe I shouldn't use blue anymore. Alright. So now we got our clean board. Let's continue. And we just have a few more names to talk about. So first we have, let's see. So we have Levine, Phoebus Levine. I'll be honest, off the top of my head, I don't quite remember when that was. And I don't have it on my little note sheet, so I apologize for that. But what he found is that by analyzing DNA, that there were equal portions of certain molecules. So he noticed that there was a phosphate group, a sugar group, and a base group found within the DNA. And when he noticed that, he also noticed that they were in equal proportions. So for every one sugar, there was one phosphate and one base. So it was all in equal proportions. That kind of hinted that, those things went together to form what we now know is a nucleotide, the monomer of DNA. Now we move on ahead. We have Chargaff. Smart dude, has his own rule named after him. We call it Chargaff's rule, So Chargaff, don't quite remember the date of this offhand. Again, I'm sorry. But what happened, sorry, I also forgot one more. Alright, so we'll get back to that one. So Chargaff we talked about this in our first video. He figured out that the number of purines and pyrimidines were equal to each other, and that led to Chargaff's rule, which says that adenine and thymine pair together, and cytosine and guanine pair together. Now they're able to do that because we have one purine and one pyrimidine, and they have different numbers of bonds. So that's how they're able to pair together. And what that essentially boils down to is helps us figure out the structure of DNA. We'll get back to that. I did forget something that was before these two gentlemen, and that was in 50, when we have Hershey and Chase. Now they did what's called their blender experiment. I was gonna, Hershey and Chase. See, just like a real classroom, sometimes we skip over things and we have to go back and improvise. Alright, so Hershey and Chase did their blender experiment, and what that was is they took a bacteriophage, which is a virus that infects bacteria. And what they were looking at is trying to solve the debate of, is DNA the hereditary molecule or is it protein? So they set off to figure this out. And what they did was they used these little radioactive tags. What it helps is, sorry, I'm a little self-conscious about puffiness in my shirt. So what it boils down to is that how a virus replicates is that it injects its genetic information into a host cell. When it does that, the genetic information hijacks the host cell in order to use the host cell to make a whole bunch more of it. So what they did was they put these little radioactive tags. Now, obviously not to scale, but they did two groups of experiments. One where they put the radioactive tags on the protein of the virus, and one where they put the radioactive tags on the DNA of the virus. So what they did was, in the protein group, they added the radioactive tags. It's mostly just so that they can figure out what it goes where if the protein is removed from the virus and put into the host cell. So they mix it up with a solution that contains bacteria, hence why we call it the blender experiment. Whether or not they actually use a blender is a really good question. But ideally, I like to pretend that they do, or at least, think that they do. So they mix them up, and then they look for the tags. So they're using their machinery, their equipment, and they're tracking where the radioactive tags are located. And what they noticed is that not a single one was inside the bacteria cells. They were all outside in the solution. Then they did the same thing, but with the group of viruses that had the DNA tagged. Mix them up, woohoo, with our blender. And then what they noticed is that in this case, the tags, the radioactive tags, were all inside of the host cells, the bacteria cells. So that showed that the viruses were injecting the DNA into the host cells, which means that had to be the genetic material. All right, so then we have Levine, then we have Chargaff, and now we have some of my other peoples, the last people we're going to talk about for history. Just want to make sure I get the dates right. All right, I don't really have the dates on there, but I remember it's early 1950s. I want to say around 1952, 1953, give or take. And there's a scientist, a young woman scientist named Rosalind Franklin. And she's using a technique called X-ray diffraction. And when she did that, she took her famous photo 51. And what this shows, now this is using X-rays, not light waves. So when we talk about it being a photo, it's not exactly like what you take with your phone. It's using X-rays, so it comes out a little bit different. But what it showed was an X pattern in the photo, again, for lack of a better term. And that X pattern hinted that there were two strands and they were twisted. So what Franklin figured out was that DNA, the structure of it, had to be a double helix. Now, with her data, Watson and Crick, who actually were two of the three people that got the Nobel Prize for figuring out the structure of DNA, Watson and Crick took her photo with the help of another scientist, Maurice Wilkins, and they created the first 3D model of DNA. A lot of times, Watson and Crick are credited with finding the actual structure, but that's just not true. It was Franklin. She figured out that it was a double helix. And then Watson and Crick figured out from her data how the 3D model should look and create the 3D model. Now, should they have gotten the Nobel Prize for figuring out DNA was a double helix? In my humble opinion, no. But unfortunately, back then, it wasn't known. Franklin died not long after from ovarian cancer due to X-ray exposure. And Watson and Crick got the Nobel Prize. However, then, the past decade or so, we've started giving credit back to Franklin for actually figuring out that it's a double helix. So she figured out the shape. They figured out the 3D model, which is still important. I'm not trying to make light of their discovery at all. It is still important because once we had that 3D model, it became much, much easier, very, very clear how DNA replication could occur, which we'll talk about on another video. I don't want to, put you guys to sleep already. So that's my brief history on DNA and how we figured out what it is. Hopefully I wasn't too all over the place. And, just a little bit of review. Miss you guys. Hope you're staying healthy amidst all this. And until next time, take care.